Thanks, Stephen, and thank you as well to the GRDC for giving me the opportunity to come and present some key findings and preliminary findings here today. So really do appreciate it as a student being able to share my research with you. There we go. So as most of you will know, and you will hopefully have all heard of subsoil manuring, and this is a technique that's been developed here in the high rainfall zone by some of the people in the audience here today. And the aim of subsoil manuring is to ameliorate or improve our hostile clay subsoils, like this one here, and increase crop yields on these soils. So subsoil manuring is the incorporation of high rates, so 10 to 20 tonnes per hectare, of nutrient-rich organic matter like poultry litter into the subsoil, usually by deep ripping. Now, since the early 2000s, there have been quite a lot of field and controlled environment trials on this research, and we have evidence that subsoil manuring improves the chemical fertility of the soil, it improves the soil physical structure, which that image is showing there, and it also enhances the biological activity. So there's evidence that it has both a soil conditioner and a fertiliser effect, and taken together, this translates to the significant improvements in crop productivity that we see in trials. But as you are also probably aware, even though trials have been quite promising, large-scale adoption of this technology is still very limited. Only a handful of growers, if that, in the high rainfall zone have adopted this. Now, as well as the more obvious barriers to adoption, like the cost of subsoil manuring and also access to machinery, there's a number of research questions and research gaps that still need to be answered so that we can understand the what, the how, the why of subsoil manuring, so we can improve the reliability and the robustness of the technique, and so we can improve the cost effectiveness. And these questions include, which factors are driving increases in yield? Is it the nutrition supplied by the amendment? Is it the fact that the subsoil is ameliorated, or is it something else altogether? Why do results vary in different seasons, in different crops and different years? And what's the expected duration and degree of response? What's the ideal method and ideal depth of placement? Is a surface applied amendment going to yield the same results as a deep ripped one, for example? And which amendments work and why? Will an inorganic amendment provide the same results as an organic amendment? And finally, do the microbes introduced via the organic amendment actually enhance the amelioration process at all? Now today I'd like to focus on questions surrounding the amendment, but first I'd just like to touch on why I'm interested in soil microbiology specifically. Now, there's a growing appreciation for the complex and interconnected nature of soil physics, soil chemistry, and soil biology. And the living soil biota is now understood to be absolutely critical to the structure, function, and productivity of soils. Microbes, including bacteria and fungi, are involved in most soil properties and processes, including decomposition, nutrient cycling, and aggregation. So all things that are hypothesised to occur after subsoil manuring and contribute to the yield response. So in order to understand the whys and the hows of subsoil manuring, we need to understand the role of soil microbiology and how the microbes link to the soil characteristics and plant growth. In order to investigate this, I'm carrying out a series of field sampling experiments and controlled environment trials. So these involve going back and testing a range of existing subsoil manuring sites, as well as carrying out some more detailed experiments in the lab. In each experiment, I'm looking at both spatial and temporal sampling to capture changes in measures after manuring over time relative to where the amendment is placed. And in each experiment, I'm taking measures of microbial community size, structure, and function, and then pairing these with soil physical data, soil chemical data, and measures of crop performance. And by doing this, I can understand which factors change after manuring, how these factors correlate with changes in the microbial community, and then what's driving the increases in yield. So going straight into the results here, from sampling of historic field trials, in this case from Yallock Estate, we know that the soil microbial community is changing as a result of subsoil manuring. So here, amongst both the bacteria and the fungi, the subsoil manured community and untreated community are significantly different. And we also know that these communities remain different for some time after treatment. So in this case here, nearly 3 years after the poultry litter was incorporated into the subsoil. Now this corresponds to the duration of yield response that we see in the field. We have sites that have recorded yield responses for 3 to 5 years after treatment. We've also learned that after subsoil manuring, the microbial community of the topsoil, of the subsoil, sorry, more closely resembles that of the topsoil. So here we see the community fingerprint of the topsoil, subsoil, and poultry litter prior to incubation. So what you're looking at here, each individual circle represents a unique species, and the size of the circle represents its abundance in the community. So after subsoil manuring, when the poultry litter is added to our dense clay subsoil, the fingerprint of the ameliorated subsoil more closely resembles that of the topsoil. And this also corresponds to observations from earlier experiments where we see changes in the soil physics and the soil chemistry of the ameliorated subsoil to more closely resemble the topsoil. For example, it becomes more fertile, more porous, better structured, and so on. In terms of the type of amendment, it appears that amendment quality or nutrition is the most important factor. So these 3 fingerprints show the microbial community of a clay subsoil after 20 weeks incubation with 3 different amendments. And you can see that in a short-term incubation at least, the 3 different amendments generate the same shift in community structure regardless of whether they're organic, inorganic, or a combination of the 2. Now, this is also regardless of whether the amendment contains carbon, which this treatment here, inorganic nutrients, does not contain added carbon. Now this is interesting and it supports the most recent yield results coming out of the Southern Farming System's Westmere trialled site, where we're seeing that the key thing that's driving yield at that site is the nutrition, especially the nitrogen of the amendment, not subsoil amelioration per se, not depth of placement of the amendment. And finally, if we compare these 3 amended subsoils to the raw poultry litter here prior to incubation, we learned that the raw poultry litter doesn't contribute any unique microbial species to the poultry litter amended subsoil. So all the same species occur, whether the subsoil is amended with poultry litter, inorganic nutrients, or wheat straw nutrients in this case. So what this is telling me is that it's the nutrition supplied to the native community and the feedstock that this community needs that's important for driving community structure, not some inoculation of microbes from the amendment. And if this is the case, and if we validate this, this opens up other potential opportunities for amendments that might have the same nutrition as poultry litter but are more cost-effective and cheaper to use. So just to quickly summarise these take-home messages for you, the microbial communities change within 2 weeks after subsoil manuring, and probably a lot quicker. I just didn't take any measurements quicker. And they remain different for at least 2 years and longer. The physical, chemical and biological properties of the ameliorated subsoil more closely resemble the topsoil. And the nutrition of the amendment and the stimulation of the native community is more important than inoculation of microbes from the amendment. And finally, it's just important to note that this is an ongoing project. I'm still working on this only halfway through, so they are preliminary findings. But as I'm going forward now, I'm looking at higher resolution techniques. I'm focusing on identifying the key microbial species that are changing here and their functional roles, and then linking those to the soil physicochemical characteristics and crop performance. Before I finish, I'd just like to acknowledge my supervisors, colleagues and collaborators, as well as the GRDC for providing additional scholarship for this project. And thank you.